Yes, welcome guys to another lesson of ours, where today we are going to talk about mutations. And previously we talked about genetics, and I hope all of us here are able to carry out genetic crosses. So mutations, I think it might not be a new word to all of us. However, it might be new to some of us, which is okay. That's why you are here and we are here to learn. Now, mutations, it is a sudden and it is spontaneous change. Sudden means it's rapid, it's quick. Then spontaneous means it occurs on its own. Sudden, it's like it's probable. You don't expect it to occur. You don't expect it to happen, but from nowhere, it occurs. And it occurs on its own. So it's a sudden and spontaneous change in the structure and composition of a gene. And if you've been attending all our lessons, you know what a gene is. You know what a chromosome is. And you know that we are what we are because of our DNA, and DNA contains genes. And the DNA is wrapped onto the chromosome. So any change in the chromosomes, any change on a gene, or any change on a chromosome, for instance, it will mean the change is on the DNA. And any change on the DNA, it means it will change the entire organism. Okay? So that change that occurs on the DNA, on the gene, and on the chromosome, it results into the change in the overall phenotype of an organism. Okay? It results into a change in the expression of that gene. Now the change might be good or the change might be bad. Okay? It might be good or bad, depending on. So examples, we have sickle cell anemia. We talked about this. We talked about sickle cell anemia. Did we talk about this? Cystic fibrosis? No, we didn't talk about it. Now with cystic fibrosis here, it is also a mutation. It is also a change that occurs in your DNA, which leads to somebody producing an abnormally thick mucus. Abnormally thick mucus. Now, with this abnormally thick mucus, it becomes hard, okay, to be released out of the trachea once it is formed. You know, the trachea lining, it is covered with mucus. And it is the mucus which traps the microorganisms, the dust and stuff like that. And then after it has trapped, then you have the cilia, the hair, okay? So if this is the trachea lining, you have the mucus that lines the trachea. And then also these cells that make the lining of the trachea, they are hair. They are hair-like. They have hair. Okay? Called cilia. Now, once this mucus has trapped microbes, dust, stuff like that, then this hair is going to beat rhythmically in a coordinated manner so that it moves this mucus together with the microbes trapped and the dust trapped. to the larynx, and then to the pharynx, so that you can cough it out, or you swallow it to be broken down. Remember, when you swallow it, the conditions in the stomach, the stomach is too acidic, the pH is around 2. You have their enzymes which are tough, okay? So they will break it down into harmless, and they render the microbes harmless. And now for cystic fibrosis, these fellas, they produce an abnormal thick mucus, okay? Due to some mutation happens, and I want to go into details. That's enough. So when you produce this abnormally thick mucus, it means when it traps microbes, it won't be able to be swept up. If it is here, it is thick, too thick, too thick. It won't be able to be swept up. So it remains here. Remember, it has trapped microorganisms. The microorganisms will multiply within this abnormally thick mucus, okay? And they become many and many and many, and they end up infecting your cells. So that condition is cystic fibrosis, and it is also caused by a mutation. Then resistance in bacteria and viruses. I think all of us here, we are good at watching news, but not only news, that canary show, canary mugume, that extension on COVID what stuff, the 4 at 9. Whenever the guy would appear, he would talk about variants of coronavirus, lambda variant, theta variant, gamma variant, alpha variant. Didn't you hear such words? Didn't you? Even if you're not good at watching news, at least you'd be there when mommy or dad is watching, and you hear lambda variant, gamma variant, alpha variant, and stuff like that. Tracy didn't. Seems you're not good at watching news. Now, the coronavirus, the Wuhan strain was one, but the Wuhan strain kept on changing up to those different variants, okay? Lambda, mu, what. Now, it's this mutation that allows these viruses to change. Even bacteria, even plasmodia, even they also change, okay? For example, let me use this. I know it's familiar. I'm quite sure all of us here have ever been in a situation where there are bed bugs, either at school, either home, or at friends. If you've never been at, but you've ever heard of, okay? Now you all know that you cannot spray bed bugs and they die, all of them. I'm sure that's what it is in your mind. You spray, some die, but after a month, they re-emerge in large numbers, even more than what you had previously. You remember, you know that? Yes. Let's leave the issue of bed bugs. You know, if anyway, you are all 2,000 kids, but this malaria drug, it has undergone, is it undergone or underwent? Whatever. Different evolutions, whereby in 90s they were using chloroquine, then camaquine, then bra, and then they developed coartem, and then from coatem, ramatem. Now from ramatem, I don't know what's there now. I don't know what they are giving now, but it keeps on changing. Have you ever wondered why it keeps on changing? It is because even the plasmodium too, it also changes. Okay? It also changes. Somebody's saying something here. Somebody's saying technology. Yes, it is technology, but why should it be changing? It's because the plasmodium, it also changes. And when it changes, it becomes resistant to the drug that you are administering at that moment. Okay? So therefore, you cannot swallow a particular drug and you get healed of you get healed of malaria. No. Because the plasmodium has become resistant to that drug. So you have to change the drug so that the drug becomes new. But as this new drug is changed, then the plasmodium, it will also undergo some mutations which lead to forming strains that are resistant to that drug. So therefore, I need to make another one. And so that's how science is. That's how things are. Then finally, we have dwarfness. Okay? Dwarfism. Dwarfness. And if you're a good person who watches movies, I'm sure you've ever seen such a guy. Now this guy, imagine, you see how tall he is. Okay? But you see where others are. Okay? You see where others are. Too short. Okay? This is a mutation. Okay? Called dwarfism. Fellow pygmies from Kasese. I'm not saying that all people who come from Kasese are pygmies, no. But there is a certain race of pygmies. Too short. Okay? I hope we don't have one here. But anyway, I'm not trying to. But they are short, okay? And it's a mutation. Okay? It is a mutation whereby, okay, let me give the science, let me talk some little. So whereby these people, they undergo mutation to form, now in your body you have what we call growth hormone. Now it is that hormone that makes you attain that maximum size of yours that you have. Okay? It's because of that hormone. Otherwise, if it's not, then you wouldn't be able to grow. You remain with short bones, and if your bones don't elongate, then you remain short. Now, these people, yes, they do produce the hormone, but they do not have the receptors for that hormone. So they have changed. The gene that produces the receptors has changed, that they no longer produce the receptor. So they remain short. So the same applies to this guy. You can see how short this guy is. So anybody who has ever seen this guy? Anybody who is a movie watcher here? Who is good at watching movies? And which movie was this? It's okay. So basically that's what mutation is about. Okay? Any change in the DNA, in the structure and composition of DNA, or you can say of chromosome, or you can say of a gene. leads to the change in the phenotype, leads to the change in the phenotypic expression. And that change is what we refer to as mutation. Like that guy, like this guy. This guy, if you're good at watching, you can see he's supposed to gain a normal height. Okay? Supposed to gain a normal height. Now there is a difference between dwarfism and being short. Now being short is normal. But dwarfism, it's a mutation. Now this guy is not short, but he's a dwarf. Okay? He's a dwarf. In that he mutated and so he ended up becoming like this. Good morning. Okay? Yes. So types of mutation, the 4 types. Can I receive some queries? So like I told you, we have 2 types of mutations. Number 1, the chromosomal mutation, and the other one is gene mutation. Somebody is saying, teacher, why? Why what? It's okay. Why what? Okay. So if this is our chromosome, you remember when we were studying, when we were introducing, we talked about what chromosomes are. Okay? They are just thread-like structures that bear DNA or that have DNA. But my drawing is not neat. Let me draw another. So if I draw this, okay, let's all mute. If you want to ask, tell me about all of them. I'm going to talk about all of them, so have no worries, okay? But they are mainly 2 types. But I'll talk about all the deletion, translocation, duplication, and stuff like that. Let's all mute. Okay? Yes, let's all mute. Now, if this is our chromosome, we know that our chromosome, the whole of this, has DNA. Actually, it is the DNA that is coiled on certain proteins, okay, called histone proteins. And you keep on coiling until when you come up with a chromosome. So any change that happens, for instance, you break down this and you lose it. Okay? Okay, I was going to rub, but I might rub everything. So you break down this and you lose it. So let me not rub, I might rub the whole chromosome. So you break this segment and you lose it. You've not lost just a segment, but you have lost DNA. Okay? Now the DNA, it has genes, and genes code for protein molecules. So it means you are losing your genes. So it will result into a change in the phenotype. We all know that phenotype means the outward expression. So the outward expression is going to be different. For example, think about sickle cell anemia. Here it is. Somebody talked about a single base substitution, and I was happy to hear about it. But if I'm to talk about it, however, this is not O level, it's A level. But since somebody talked about it, then it's okay. Now, a gene is made up of 3 bases. Okay? 3 bases. Now, these three bases, they are the ones that determine the amino acid. And you know, in digestion, you break down proteins, you form amino acids, okay? As the building blocks, as the single unit that can be formed when proteins are broken down, as the end products. In fact, that word is better. So also when you want to form a protein, then you add these amino acids together. Okay? You add these amino acids together to form a protein. And what happens? It is this DNA that codes for a given protein that is going to be formed. For instance, if it is hemoglobin, you know hemoglobin, it carries oxygen, and we've talked about sickle cell anemia. Yes? So if it is hemoglobin, the hemoglobin is going to be coded by DNA. Maybe this segment of DNA, you see? Now this segment, it has very many organic bases. And these organic bases must be maintained constant. They should not change. They should not change at all. Now, if anything happens, we are going to see agents of mutation. So if something happens and you change this base, you change this, maybe where there was G, you remove G, and maybe you put something like, okay, C. Let's put C. Don't ask me what A is. Don't ask me what G is. Don't ask me what T is. You don't need them. But let's use them to illustrate our. So if something happens where there was G, you put C. Maybe here we put C. Everything is going to change. Everything. And this protein is the protein which was hemoglobin, which was normal hemoglobin, it is going to change into some other protein, some other stuff. Okay? And this change is manifested as a mutation. If it is sickle cell anemia, then it becomes sickle cell anemia and somebody cannot survive. Can you imagine? Yes. So that's how mutation is. So now, depending on where this has happened, if it is on the DNA, then we call it gene mutation. But if the change occurs on the entire chromosome, then it becomes chromosomal mutation. Any question, guys? Any queries? Please feel free to ask any question. I'll be able to attend to it. Okay? Yes. So have no worries. So let's talk about chromosomal mutation. Let's see what chromosomal mutation is. Now, with chromosomal mutation, it's the sudden change occurs on the structure of the chromosome. It's a chromosome that changes. Now, we all know that chromosomes carry DNA. By now, that should be a basic. Okay? Yes, and I'll try to elaborate that. So you all know that chromosomes carry DNA. So anything that occurs on the chromosome, then it means it is occurring on the DNA. So any change on the chromosome, it means the DNA is changing. And we all know that the DNA is the masterpiece. If you know how to lay briquettes, how to lay bricks, how to, you know that there is that, how do you call it? That car box or that car table or that masterpiece, where you get your mud, where you get your sand, and you put it inside. Okay? and you come out with a briquette. Did I say briquette? You come out with a paver, you come out with a brick, you come out with stuff like that. Stuff like that. Okay? Yeah. Now, okay? Samanya Santiago, from where? It's okay, you can. It is okay. You can ask from where? From chromosomal mutation?
Yes, ma'am.
Okay, it's okay. But you know what a chromosome is?
No,
You don't. Did you attend our very first lesson?
no teacher.
Oh, it's okay. It's okay. Have no worries, okay? Have no worries. Okay, let me try to explain in details, okay? There is nothing to worry about. So in your body, you have there what we call you have cells. Maybe let me use some yellow pen. Red in red, it's not sex at all. So you remember senior one when you drew an animal cell? You remember that? Yes. Where we said that that's an animal cell. And then this one, this is a cell membrane. Okay. Then we said this is cytoplasm. And then we said this is a nucleus. You remember? Okay, that's good. Chromosomes are thread-like structures bearing genes. Yeah, that's good, Faith. Yeah, that is true. They are thread-like structures that do have, they bear genes. Now, from this basic structure of a cell that you studied in senior one, if we look at the nucleus, are you seeing the nucleus? That's good. If we try to magnify the nucleus more, we shall find that in the nucleus, you have there tiny, very small structures. Okay? Inside the nucleus, very small structures. Now, these small structures, they are the ones that we call chromosomes. Okay, let me write the whole word. They are the ones that we call chromosomes. Is it okay? Now, also when we magnify these chromosomes, to give you some simple understanding, if we magnify these chromosomes, then we shall form something like this. This, it is highly coiled. And this one actually is your DNA. Now it is this DNA that determines everything about you. And I usually say that you are what you are because of your DNA. Look at yourself. Look at yourself. Everything that is about you. Everything about you is because of the DNA. And by the way, you are half your dad and half your mom, whereby you get half DNA, maybe this one from mommy, and half DNA from daddy. And so this DNA, when the two join, now they start coding for proteins. And they are proteins that will make you. They are proteins that will make you. If you are tall, then it's because you have genes for tallness. And what are genes? Genes, they are segments on DNA that code for particular proteins. For example, you may find from here to here, they code for protein A. And then from here to here, maybe this is a segment that codes for hemoglobin, for example. Okay, hemoglobin, for example. Then from here to here, maybe this is the segment that codes for skin color. Okay, let me say melanin for some simple understanding. Now, anything, now if this segment is the one that codes for hemoglobin, if anything happens and there is a change, okay, in this segment, then you won't form normal hemoglobin. If anything happens and there is a change in this segment here, then you won't form normal melanin. Now that's when somebody is going to become an albino. So guys, let's all mute. If you are not going to say something, it's better we keep mute, okay? Yes. Not so guys. So is this some good basic? Is this some good explanation? Yes. Yes, so feel free to ask anything. Feel free to ask anything. I'll be happy to attend to your queries. So now, the change, anything, something might happen on the chromosome here. That change, we are going to see agents of mutation. We are going to see things that cause our DNA to change. So if something happens on the chromosome, and the chromosome, we have said that chromosome carries DNA, and that one becomes chromosomal mutation. Or if something happens on the DNA, okay, you can see DNA, not necessarily chromosome, then that one is called gene mutation. We are going to see, so have no worries, okay? Yes, so I hope this illustration is ready, okay. Somebody is asking that when I have a brother and a sister, why doesn't my DNA and theirs, why doesn't it show 100% but 25%? Maybe if I'm to understand your question, you are asking that why doesn't your DNA and that of your brother match? Why isn't it 100%? Okay, but rather 25%. That's the question. Now, to answer that, I'll take you back to the genetic process that we've been carrying out. Okay, we saw if maybe, for instance, dad is capital A, small a, okay, and mom is capital A, small a. If you cross this, now this might be your brother, capital A, this is your brother. And if you do this, this is also your brother, capital A, small a. Maybe this is your sister, capital A, small a, and maybe this is you, small a, small a. Now these two are the same, and their DNA is the same. Now, you know that fertilization is random. It's probability. Any gamete can fuse with any gamete. So there is chance of forming any of these four, any of these two. These three, sorry. So if there is a chance, you may find, yes, you are brothers, but you are different. This one is like this, and you are here. You are brothers, but you are different. because of the genes that are carried by the gametes. And depending on which gametes will fuse, it will determine which offspring will be formed. So let's talk about chromosomal mutation and gene mutations. There is a lot we can talk about them. So we said if a change happens on the chromosome, then that one, it will lead to change in the DNA. And if you change the DNA of an organism, then you have changed the phenotype of that organism. So we have types of chromosomal mutations, and they are the same types. They are the same as types of gene mutation. So number one, we have deletion, and these are just English words. Deletion, you delete. Delete. If I say delete my contact from your phone, what do? You long press a contact, and then you click delete. And you permanently remove my contact from your phone. Not so. That's brilliant. That's brilliant. So with the deletion, a piece of a chromosome is broken off and lost. Therefore, the chromosome becomes shorter than original one. Have a look at this. You see? Where is my pen? It's here. Have a look at this. I think this. So this is our chromosome. Let me change to red. I think red is. So this is our original chromosome. And something happens. In your mind, you should be asking, what's that thing? What is that thing? What is it? What is it? Okay? That's why you should be asking yourself in your mind, what is it? What's that exact thing? So something happens and you accidentally delete this segment here. I think so that our new chromosome does not have that segment. I think now you are not just deleting a chromosome, no, but by deleting this, you are removing DNA. You are removing DNA. And my dear, DNA, you don't know how much DNA you've removed. You don't know how much proteins you have lost. So as a result, this chromosome is different from this. And so the proteins formed by this, they will be different from proteins formed by this. And hence the phenotype is going to be completely or totally phenotype, totally different, which we refer to as mutation. Is it okay, guys? Feel free to ask. Feel free to ask anything. I'll be happy to attend to it. So basically, that's how simple, that's how cool deletion is. Let's talk about others. Let's see. So, is it Pavin? Yes, it's Pavin. I hope you now understand what deletion is. So deletion, it is type. It is how actually chromosomal mutation can occur.
Yes, teacher.
Then, yeah, that's good. That's good. Can you change DNA of a grown-up person? No, you cannot change DNA. You totally have no impact on the DNA. Then another way how mutation can occur on a chromosome is inversion. Invert. Any English around? Any English? Who is English? You know what inversion means? Yes. Yes. Inversion. You mean? Okay. So with inversion, this is just English. There is nothing hard, guys. A piece of a chromosome breaks and joins on the different side of the same chromosome. A piece of a chromosome breaks and joins on the different side of the same chromosome. For example, have a look. So if this is my chromosome, you see, the whole of this is my chromosome or chromatid. Okay? You can see the order is A, B, C, D. Okay? This is A, B, C, D, E, and F. Now, if this A breaks from this or from this side, okay, and joins F here, or alternatively, A can break from this segment and F2 can break from that segment, and F joins B and A joins E, and we form this new chromosome. Okay, you can see, you see where A is. Do you see that A is joined to E and F is joined to B? Do you see that the order has changed? Yes. Now this is what we call inversion. And it's not a mere change of letters. Let me quote Museveni's words too. Seven said it's not a mere change of guards. It's not a mere change of guards, but a fundamental change. So even here, it's not a mere change of letters, but rather you are changing DNA. You are changing DNA. And any change of DNA, you get totally new combinations. And it's these new combinations that will result into new phenotypes. For the new members, phenotype means the outward expression of the gene. Okay? And these new phenotypes, they are the ones that we call mutations. Master, is it true that all chromosome structure mutation begins with a break in the DNA? No, it is not true that the DNA must break. We are going to see other cases, the gene mutation, where DNA is not breaking. Okay? But maybe the good word you are supposed to use, the DNA must change. Okay? The DNA changes. Now, if a change occurs on the DNA or chromosome, then it will result into a mutation. So that was some brilliant question, guys. Then another type of chromosome mutation is duplication. If I say, can you duplicate yourself? What do? You make another you. I don't know how you make another you, but that's what you do. You make another you. You make another you. So with the duplication, a chromosome gains a piece from another chromosome of the same type and becomes longer. You can have a look at this. You have B, And if this chromosome gains another B of the same from another chromosome, this the new chromosome will be like this: A, B, B, C, D, E, F. Can see? Now this is duplication. And it's not just duplicating letters, but you are duplicating DNA. So you are going to form new combinations of genes. And because of these new combinations, then you form new proteins, which will result into the change in the phenotype. Like we said, phenotype means the outward expression of the gene. Now, if the phenotype changes, then we shall say that mutation has occurred. Then we have translocation, I think. Now with the translocation, let me rub here. Now with the translocation, a segment breaks from one chromosome and joins another chromosome. Okay? You can have a look at this. You see, this is A, B, C, D, E, F. This is one chromosome, or chromatid, okay, chromosome. This is one chromosome. Then G, H, I, J, K, L. This is another chromosome. Okay? But now if this one breaks from here, okay, and this one also breaks from I, between I and J, can see that? And it is translocated from here to another chromosome. And this one, JKL, is also translocated from here and it attaches here. These are the new chromosomes that will be formed. You can see A, B, C, instead of D, E, F, now we have JKL. Instead of G, H, I, J, K, L, you have G, H, I, D, E, F. So you are exchanging the whole segment. Okay? What does that mean? The whole DNA is being exchanged. And so impacts on the phenotype of the organism. So if the phenotype changes, then we shall say that mutation has occurred. Now with the gene mutation, the change occurs on the chromosome. Somebody is asking, do they all result from an error in cell division? Wow, this is some good question. Some good question. So somebody has some knowledge as far as cell division is concerned. Actually, that's our next topic. If we finish this, we shall go to cell division. But these ones, they don't occur due to errors in cell division. We are going to see causes of mutations. You are trying to answer my question. Okay? The question that I gave you. That think about which things happen, which issues happen that lead to those changes. Think about those mutagens. Hey, I've even given you what to research about. Okay. So sudden change in the structure and amount or in the structure of a gene. And a gene is that segment on the DNA that codes for a particular protein, and proteins make up the phenotype. So if the change occurs on the DNA, not chromosome, then we shall say that gene mutation has. Now the types, they are the same as what we've been saying. They are the same. However, the other ones were occurring on a chromosome. Now here they are occurring on DNA. That's the difference. That's the only difference. But the rest is just the same. So with the deletion, you delete a base. So if this is, okay, where is the pen? Like I told you, okay, yellow, I want, let me use yellow. It's the one which will be favorable here. So if this is DNA, I say DNA is made up of very many bases. We shall see it. No, we shall not see it. But when we go to A level, that's when we shall see. Then you have G, you have C, you have T, and stuff like that. Okay? Stuff like that. Now, if anything occurs and you delete from here, or you delete this segment, the DNA is going to change from that one to G, and then to C. You've deleted T, then you have A, then you have T, and then you have. So these two are different and they will code for totally different protein. And so there will be a difference in the phenotype. Then with duplication, same thing. This segment or this one duplicates and you form another C. Because you've added, okay, a different base, then the combinations of genes, the combinations are going to differ from this one, which will result into change in the phenotype. Translocation, same thing. Segment on DNA breaks from here and joins another segment of the different DNA. Then substitution. Somebody saying something? If mother carries gene mutation in her eggs, can she send it to her? You cannot carry G. Okay. Yeah, you can. These mutations are inherited. That's why an albino will have chances of giving birth to an albino. That's why if you have sickle cells, you have chances of giving birth to somebody with sickle cells. So they are inherited. Okay? Yes. If you want to ask your question, please raise your arm. I'll pick you up. If you don't, use the chat box to avoid those echoes and stuff like that. Then with substitution, now this one, you remove base G and put another base. Okay? It's like you get G, you remove it and put another one, maybe C, maybe T. Now that is substitution. Even a substitution of one single base, my dear, it's enough. Actually, let me give you this. For sickle cell anemia, it is just one single base that is substituted. You remove C and put G. Just one single base, not even deleting, but substituting. Okay? Do all gene mutations cause health problems? Some mutations are good, some mutations are bad. Depends on the impact. Okay? Yes, but most in humans, they are bad ones. They are bad. But anyway, some mutations are good, others are bad. Okay? Yes. So you just replace one base with another base, and what is formed is going to be totally different. The hemoglobin that is formed, we saw that is sickle-shaped in a little oxygen, and that sickle-shaped hemoglobin has issues. Okay? It has issues, like how we saw. But it is just substitution of one single base. So really, mutations are good, but on the other hand, they are bad. So can we, causes of mutations, can we drop some? Yes, that's the question I gave you. Can I receive some suggestions? Can I receive some answers? Okay. Yeah, thanks guys for the answers. Somebody saying high temperatures. Okay, that's good. That's fair. High energy particles. Yeah, that is. Now this is correct. Such as you can. Exposure to specific chemicals and radiations. Yeah, that is. Oh, you guys can make some research. Blood donations. Blood donations don't cause mutations. Good, thanks for that. Yeah, certain chemicals such as alpha gas, sorry, mustard gas. Yeah, that's good. That is good. That is good. And then alpha and beta what? That is good. You guys, your answers are amazing. They are really good. They are really good. Trust me. So let's see. Ionizing radiations. Oh, okay, Angel. Where did you get that? Accidents during normal chemical transactions of DNA. You people, you can make research. Okay, your answers are still flowing. Go ahead. High energy radiations, e.g. X-rays. Yeah, thanks, Florence. That's good. That's good. Yeah, I think you answer. Okay. Okay, let's see. That's good. That's good. So number one, okay, people are still dropping answers. Ionizing radiations. Okay, that's good. So number one, mutations are caused by substances that we refer to as mutagens. I say this word, and I think I gave you a hint. But they are called mutagens. And these include high temperatures. Yes, if you expose it to high temperatures, you cause your DNA, when DNA fails to accurately, I'm reading someone's answer. When DNA fails to accurately, okay, maybe, okay, let me finish it for you. To accurately, replicate errors in DNA replication can lead to mutation. Exposure to high energy electromagnetic radiations, e.g. UV light. Okay. Yeah, this is good, guys. This is really good. It is impressive. In fact, I should always let you explain, and then I just come to give concluding remarks. Maybe we shall try it next. I leave you, you unmute yourself, talk some science. Then another one, unmute, talk some science. And then I come to conclude. Somebody is saying chemical mutagens like smoking. Okay. Let's see some examples of mutagens. High temperature. Yes. Chemicals such as mustard gas, colchicine, and caffeine. Now this is an nerve gas. This one prevents anaphase. We are going to see what anaphase is. And this caffeine yes, so these chemicals can lead to changes in the DNA and changes in our chromosomes, like how we saw those changes. Then high energy containing particles, such as alpha and beta particles, okay? The X-rays and gamma rays, they do contain high energy. That's why it's at times not good to go for X-ray scanning. But anyway. So they are high energy containing particles. Somebody is asking, can it be caused by an impact on the protein sequence? People, where are you getting those words from? Yes, any mutation leads to the change in the protein sequence, okay, which leads to the different phenotype that is formed. Okay, that's good. Have a protein sequence, that's enough for you. Don't go beyond that. What of taking unprescribed drug for a long time? Drugs, they won't lead to mutations. But they will lead to other complications such as drug resistance, damaging your liver, and stuff like that. So it's not good to self-medicate. It's not good. So that means, so these are questions, So that means those who go for what? X-rays face mutations? No, they don't. In the hospitals, the X-rays are controlled, but errors can happen. Chemical mutagens and stuff like, yeah, that's good. Then high energy radiation such as X-rays, gamma, and UV light. Also exposure to sunlight, direct sunlight, it's not good. Okay, it can lead to mutations that you won't like. Errors during DNA repair, yeah, that's good. Hey, you people, you've been interactive. You've been so cooperative as far as causes of mutations are concerned. So can we do the same for variations? Let me receive some suggestions as far as variations are concerned. So what are variations? What are they? Okay, so now variations. I will illustrate it as this. But is time really going to be enough for us? Maybe I'm going to give you an overview, then you go and read about variations. Time won't be good for us. I have a lesson, there is a lesson that is going to be taught using the same Zoom, so it won't be good for us. Now, variations. All of us, we are all human beings, right? Yes, good. We are all human beings. But Teacher Emma is Teacher Emma, Aaron is Aaron, Angel is Angel. Who else? Joan is Joan, Faith is Faith, Chagaba is Chagaba, Otiri is Otiri, Pavin is Pavin. You cannot see Teacher Emma and call him Joseph. You cannot see Joseph and you call him Pony Mary. And you cannot see Pony Mary and you call her Ruga. Not so. I think that one is quite obvious. That one is good. Somebody is dropping something in the chat box. Someone is saying immigration of genes. Okay. Now, what do you base on to say that this is Teacher Emma, this is Ruga, this is Chagaba, this is Angel, this is Katende, this is Aaron? What do you base on? Even when we talk about chicken, even when we talk about, okay, let's talk about the chicken, the poultry farm. You are in a poultry farm of 1,000 chicken, or chickens, or hens, whatever. Somebody is saying names, no? Leave names. You are in a farm of 1,000 hens. Somebody is saying phenotype we base on that. Yeah, that's good. The appearance of the color, good. Color, good, 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 good. You base, somebody wants to base on the color. That's good. So you are in a farm of 1,000 hens. If you say, I want that, you choose specifically, I want that hen. The customer, if the customer comes, he or she will be like, I want that hen. Then if it mixes with others, he will still be like, no, it is this one. How are you able to tell that it is this one, not that one? Think about that. That's where we shall begin in our next lesson. Somebody saying size, just, it's an assignment. Go and read about variations based on what I've just given you. Okay? Read about variations. That's where we shall begin in our next lesson. And I feel if you do some reading, it will be interesting. You will really enjoy the lesson. Meanwhile, don't forget to subscribe and don't forget to always watch our previous lessons. Let me send you, I sent you the link. Maybe let me send it again. Let me send it again.